Ashkelon, southern coast of what is now Israel, 1997. An archaeologist from the Leon Levy expedition is on her knees in a burial chamber that has been sealed since 850 BCE. Around her, 4 infants. No grave goods, no ceremony, nothing that suggests anyone expected to find these children here or wanted anyone to. And then, tucked beneath the skull of one of them, a small ceramic jug, intact. And when the lab got to it, it smelled, faintly, of Aegean-style perfume oil. That jug should not have been there. Not in that burial, not in that century. Doctor on that coast, not in that century. Dr. Michael Feldman had spent years working on exactly this kind of question at the Max Planck Institute for the Science of Human History in Germany. In 2019, her team published something the field had been waiting for: genome-wide data from 10 individuals excavated across 3 distinct time periods at Ashkelon. What the data showed was this: the people buried in the early Iron Age carried a genetic signal, roughly 14% Southern European ancestry, that was completely absent from the Bronze Age population that came before them. And then, within 200 years, it was gone again, absorbed, vanished back into the Canaanite population surrounding it. These were the Philistines, and they had crossed the Mediterranean from the Aegean world around 1200 BCE. And yet, the data said the exact opposite of what most people assumed about them. The people the ancient Israelites feared most, fought longest, and wrote into scripture as their defining adversaries, they came from Greece. And within 2 centuries, their genes had disappeared inside Canaan. The enemy was absorbed, completely. Here is the fact that changes everything before this episode even begins. The Philistines are not the story. They are the frame. Because what happens to a Canaanite Levantine population that survives invasion, displacement, intermixing with sea peoples from the Aegean, and then watches its nation destroyed, first by Babylon, then by Rome, is not a story of preservation. It is a story of transformation so total that the question, what are the Jewish people descended from, does not have a single answer. It has 4 women, and not one of them lived in Canaan. In the Philistine cemetery at Ashkelon, archaeologists found an adult male buried with an iron knife and 2 ceramic bowls, unmistakably Aegean in style, both made, the analysis confirmed, from local Canaanite clay. He had arrived from somewhere else. He was already becoming someone else before he died. That is where this story begins. A population migration reshaped the genetic foundation of what would become the world's largest Jewish diaspora. They left no written name. They left no kingdom. They left 4 mitochondrial sequences and roughly 3.5 million living descendants. Before you watch what comes next, where did you think the maternal ancestry of Ashkenazi Jews originated? Most people have a strong instinct about this. Drop it in the comments, because the answer is going to surprise you. 4 maternal lineages. That is the finding. Not a dozen, not a broad Levantine pool. 4 distinct mitochondrial DNA sequences account for roughly 40% of all Ashkenazi maternal ancestry. Every Ashkenazi Jewish person alive whose maternal line carries one of these 4 sequences descends from one specific woman, a single individual who lived approximately 2,000 years ago. 2,000 years is 66 generations. 66 women, each passing a single identical mitochondrial sequence to her daughters, unbroken. From a Roman street to your family tree. Professor Martin Richards had spent years at the University of Huddersfield working through this exact problem. In 2013, his team published an analysis of 3,500 Ashkenazi Jewish mitochondrial genomes. They identified the 4 founding lineages: K1A1B1A, K1A9, K2A2A1, N1B2. They estimated all 4 arose approximately 2,000 years ago. And then they mapped where those lineages showed the highest frequency matches in the modern world. Not the Middle East, Southern Europe. Specifically, the Italian peninsula. For decades, the working assumption in Jewish genetic research was that Ashkenazi maternal lineages reflected a migration from the ancient Levant into Europe during the Roman diaspora. Middle Eastern women following their communities west. Richard's data turned that assumption over completely. The 4 founding mothers appear to have been local European women, almost certainly converts, or women from communities that joined Judaism during the period when Jewish communities in Rome were actively growing, before the 4th century CE, when Roman law made proselytization illegal and the window closed. Between 100 BCE and 400 CE, Jewish communities in Rome were among the fastest growing in the ancient Mediterranean. The historian Josephus, writing in the 1st century, estimated their numbers in Rome in the tens of thousands. And the inscriptions in the Monteverde Catacomb, one of Rome's oldest Jewish burial sites, are written in Greek and Latin, not Aramaic or Hebrew. These were Roman-born Jews, not immigrants, not arrivals. People who were already there, already local, already choosing to be part of something that was not yet old enough to feel ancient. Some of those people are the source. The comfortable assumption that Jewish maternal ancestry runs unbroken from the ancient Levant has a crack in it now. But here's where it gets strange, because one of the 4 founding lineages, K1A1B1A, the single most common Ashkenazi maternal haplogroup carried by roughly 19% of all Ashkenazi Jews alive today, shows its highest density not in Italy, not in the Levant. It shows it in a geographic arc running from eastern Anatolia through the Caucasus into Central Asia. And that arc points somewhere very specific, somewhere very contested, somewhere that one of the most politically charged theories in the history of Jewish genetics claimed to have already answered. The Caucasus signal. That is what needs explaining. K1A1B1A, carried by nearly 1 in 5 Ashkenazi Jews, has its highest phylogeographic density in a region where no ancient Jewish community is documented at the relevant time. And if you've spent any time in this field, you already know what theory that's supposed to support. The Khazar hypothesis, a Turkic kingdom in the Caucasus whose elite converted to Judaism in the 8th or 9th century CE, a theory used for political purposes across the ideological spectrum, and a theory the genomics have now tested directly. Dr. Doron Behar at the Rambam Healthcare Campus in Israel published genome-wide autosomal data in 2013, modeling Ashkenazi ancestry against every plausible source population. The result was clear: no Turkic Khazar signal at any meaningful frequency. The model that fits is Middle Eastern and Southern European, not the Caucasus steppe. The Khazar hypothesis, as a dominant origin theory, is genomically ruled out. But ruling it out as a primary source is not the same as erasing the Caucasus signal. K1A1B1A is not Turkic. Its distribution doesn't match the Khazar model. What it matches is older, possibly by millennia. Whatever population carried this lineage into the Ashkenazi gene pool predates any name we currently have for them. We don't have a site, we don't have a century. We have a mitochondrial distribution that reaches toward that region and cannot be made to reach past it. In 838 CE, the Khazar Khagan sent a letter to the Byzantine emperor requesting Christian missionaries. He received them, sent them home, then asked for Jewish scholars instead. The conversion is documented. Its genetic trace, if it exists at all, has never been isolated. What we have is an anomaly with no name attached to it. 2 independent methods have dated the Ashkenazi founding bottleneck to 2 completely different centuries. The gap isn't measurement error. It is evidence of an event, possibly 2 events, that the research has not yet fully named. The bottleneck was not one event, and it did not happen when everyone thought. Dr. Shamam Waldman and Dr. Shai Carmi at Hebrew University of Jerusalem spent years building toward a finding that rewrote the standard timeline. In 2022, their team published ancient DNA from 33 individuals excavated from the Erfurt Jewish cemetery in Germany, a 14th century community, and the data stopped the field cold. The medieval Jews of Erfurt were genetically more diverse than modern Ashkenazi Jews. More diverse, which means the population contraction that shaped the genome of every living Ashkenazi person did not happen before Erfurt. It happened after it. At least in part, it happened because of what happened to communities like Erfurt. And what happened to Erfurt is documented. November 21st, 1349. The entire Jewish community of the city, approximately 100 people, was killed or expelled. The accusation was that they had caused the Black Death by poisoning wells. That accusation was false. The killing was real. Within years of that night, 3 of the individuals whose DNA was sequenced in Waldman's study had been buried in the Erfurt cemetery. The Erfurt treasure was discovered in 1998, a hoard of silver, jewelry, and coins hidden inside a wall. Someone had placed it there expecting to come back for it. No one came back. Among the objects, a woman's ring inscribed in Hebrew. The translation? Blessed be the one who comes. This isn't just ancient history. This is in your DNA right now, if you carry Ashkenazi ancestry. The standard narrative had always placed the Ashkenazi founding bottleneck in the early medieval period, small settler communities moving from the Roman world into the Rhineland around 800 to 1000 CE. Waldman's data doesn't erase that founding moment. It reveals there was a second narrowing, later and more severe, occurring precisely during the decades when Ashkenazi communities across Europe were being systematically destroyed. The genome did not merely record persecution. The genome was partly created by it. The genetic diseases carried at elevated rates in Ashkenazi populations, Tay-Sachs, BRCA mutations, familial dysautonomia, have a causal chain that runs, at least in part, through a specific autumn in a German city in 1349. That chain begins with a founding population, an original group of settlers, and the DNA is now pointing directly at who those settlers were. What the data has confirmed, across multiple ancient sites, across multiple independent research groups, is that the founding Ashkenazi population shared the same ancestral origin, the same Bronze Age source, as the very people Scripture records as their most ancient adversaries. The enemy was never foreign. Tel Megiddo, Northern Israel, 2019. An archaeogeneticist from Hebrew University is holding a fragment of a 3,400-year-old human femur. The individual was Canaanite, Bronze Age, pre-Israelite, from a civilization that existed long before the events recorded in any surviving scripture. And the results on her screen are showing something the field has been debating for 2 decades. The genomic distance between this Canaanite individual and a modern Ashkenazi Jewish person is, in certain autosomal components, smaller than the genomic distance between that same Canaanite and a modern Lebanese Arab. Dr. Waldman Carmi and Dr. Shamam Waldman's team at Hebrew University published this in Cell in 2020. Their analysis covered 73 Bronze and Iron Age individuals from Megiddo, Khazar, and Ashkelon, the core cities of the Canaanite world. What it showed was that the Canaanite population contributed substantially to multiple modern Levantine groups, including Jewish populations across diaspora communities. The Canaanites weren't replaced. They weren't erased. They became, in part, the genetic substrate of populations that would define their identity as distinct from, and often in opposition to, the Canaanite world. Everything I just told you? Hold on to it, because it's about to mean something completely different. The Canaanites were not the enemies of the ancient Israelites in the way the story was told. At the population level, they were the same people, separated by religion, by political identity, by which hill they farmed. Not, in the deepest genomic sense, by ancestry. Now, here is what this episode has been building toward. The 4 women who are the direct maternal ancestors of half of all Ashkenazi Jews alive today appear by genomic evidence to have been Italian women, very likely converts in Roman-era Jewish communities, not Levantine origin descendants of the ancient Israelites. Meanwhile, the paternal Ashkenazi lineage, the Y chromosome haplogroups J1, J2, and E1b1b, traces to the ancient Levant with high confidence. The Canaanite world, the ancient Near East. 3,000 years of documented ancestry running through the male line. The genome of an Ashkenazi Jewish person is, simultaneously, Roman-Italian on the mother's side and ancient Canaanite Levantine on the father's side. The story that has always been told as a single line is, genomically, 2 completely different stories. Fused. In 70 CE, following the Roman destruction of the Second Temple, Jewish men were dispersed throughout the Roman Empire, as soldiers, as slaves, as traders, as refugees. The women they eventually partnered with in Rome, in Carthage, in the Rhineland, these were the mothers. Their children became the founders. Their mitochondria became the inheritance that roughly half of all Ashkenazi Jews alive today carry in every cell of their bodies. And the longer the genomic timeline extends, the more European the maternal signal becomes. Not less, more. But what does European mean when the Europe in question is 2,000 years old? The best current genomic model places all 4 founding Ashkenazi maternal lineages in Roman-era Jewish communities in Italy, most likely Rome itself, between the 1st and 4th centuries CE. The Trastevere district of Rome held one of the largest Jewish communities in the ancient world, tens of thousands of people, largely Latin-speaking, substantially integrated into Roman civic life, and, critically, known from Roman sources to have been actively engaged in conversion during a period when Judaism was still openly welcoming of converts. Waldman and Carmi's 2022 cell study modeled the ancestry of the medieval Erfurt community and found approximately 65% of their ancestry aligned with Southern Italian and Greek populations, and approximately 19% with Levantine populations. The Levantine ancestry was concentrated in the paternal lineages. The maternal signal pointed south and west, toward Rome, toward the Trastevere quarter, toward women whose names the inscriptions in the Monteverde catacomb record only as wives and mothers, who lived in peace. For a moment, the story resolves. 4 women, Rome, 2nd century CE, daughters of a city that was already ancient when they were born. They chose to join a community that was being watched, periodically expelled, and already carrying centuries of displacement in its prayers. That answer is satisfying. You can feel the ground becoming solid beneath it. Hold on to that feeling, because the ground is not solid. In 2025, a team applying new local ancestry modeling to Ashkenazi genomes found something the Italian origin answer cannot fully account for. Approximately 7% of Ashkenazi ancestry carries a signal inconsistent with any known Italian population from the Roman period. The closest match in current databases is a population called Iran-Chalcolithic, the ancient inhabitants of the Zagros Mountains, a population that no longer exists as a distinct group anywhere in the modern world. The early data suggests the women we've been calling Italian founders may have been carrying ancestry from a population already ancient when Rome was young. This research is preliminary. The field has not settled it, but the implication is clear. The Roman-Italian origin is not the final answer. It is a layer. Beneath it is something older and nameless. The founding of Ashkenazi Jewry is not a point in time. It is a stack. And somewhere in that stack, in the immune memory encoded by what happened in 1349, is evidence that the body itself has been keeping records the history books forgot to write. There are roughly 19 recessive genetic disorders that appear at elevated carrier rates in Ashkenazi populations: Tay-Sachs, Gaucher disease, familial dysautonomia, BRCA1 and BRCA2 mutations, Canavan disease. If you've had Ashkenazi genetic screening done, you've seen the list, and the standard explanation is the founder effect: random genetic drift in a small population, certain variants amplified simply by chance. That explanation is accurate. It is also incomplete. Dr. Shai Carmi at Hebrew University, working with colleagues at Columbia, published in Nature Communications in 2014 the most detailed full genomic sequencing of Ashkenazi Jewish individuals completed at that point, 128 people. The study placed the effective founding population at fewer than 350 individuals, and the bottleneck timing was consistent with 600 to 800 years before the present, placing it squarely in the Black Death persecution period. But the study also found something beyond the founder effect: evidence of positive selection acting on certain genomic regions. Some variants did not reach their current frequencies by chance alone. Something else was happening. This isn't just ancient history. This is in your DNA right now. The genetic variants flagged as elevated risk in Ashkenazi screening panels are not errors in the genome. They are a census of a catastrophe. They count who survived. Each elevated carrier frequency marks a moment when the population was so small that chance, and possibly selection, amplified certain variants to a frequency that now affects medical outcomes for living people. There is a hypothesis, contested but not refuted, that the Tay-Sachs carrier variant, which causes fatal disease when inherited from both parents but causes no illness in someone who carries only one copy, may have provided some protection in heterozygous form against specific pathogens, possibly tuberculosis, possibly plague. The evidence is not settled, but the question it raises is real. What if what looks like a disease risk is also, at the population level, a record of what it cost to survive a specific winter? The bottleneck did not damage the genome. The bottleneck is the genome, and written inside it, at the molecular level, is a message that says, "We were here, we nearly weren't, we survived." And the paternal line, the Y chromosome, tells a completely different story about how far back that survival actually runs. There are three things the paternal DNA evidence revealed. The third one is the one that changes everything. The Y chromosome haplogroup J1p58, specifically the subclade carried by a large proportion of Kohanim, Jewish men of traditional priestly lineage, has been dated by mutation rate analysis to approximately 3,000 years ago in the southern Levant. This is what's known as the Cohen Modal haplotype, first described in 1997. Dr. Michael Hammer at the University of Arizona expanded this analysis in 2009, working with Kohanim from both Ashkenazi and Sephardic communities, groups separated by geography for centuries. Both groups shared a common patrilineal ancestor estimated to have lived between 2,650 and 3,180 years ago. That places the origin in the early Iron Age Levant with high confidence. Across the diaspora divide. So, the maternal DNA says Roman Italy, 2nd century CE. The paternal DNA says ancient Canaan, roughly 1000 BCE. Both signals are real, neither cancels the other. This is what the episode has been building toward, a genome that carries 3,000 years of Levantine paternal ancestry and 2,000 years of Italian maternal ancestry simultaneously. And together, those two things produce something no single origin story ever predicted. A population whose genetic diversity is the record of its own survival strategy. The diaspora did not disrupt the genetic record. The diaspora preserved the paternal record through the specific social mechanism of patrilineal transmission of priestly and tribal identity, while allowing the maternal record to evolve with every community the diaspora touched. The genome was not preserved despite displacement. It was partly built by it. In the Samaritan community of Nablus, approximately 800 people, the last remnant of the ancient northern kingdom of Israel, Y chromosome haplogroup J1 runs at roughly 58%, the highest concentration of that lineage in any living population on Earth. The Samaritans did not move. They did not merge with larger populations. Their paternal line is the oldest continuous genomic record of the ancient Israelite North, and most people outside of genetic research have never heard of them. 350 effective individuals. That is the number. That is the third thing. The entire Ashkenazi population, 10 to 13 million people today, descends from a community that, at its minimum bottleneck point, numbered fewer than 350 effective individuals. One plague season, one winter that did not break, one pogrom the survivors couldn't outrun. And none of them exist. 350. Not 350,000, not 3,500, 350 effective individuals, the number Carmi's team calculated as the minimum founding population of all Ashkenazi Jewry alive today. In a medium-sized American high school, that is roughly the junior and senior classes combined. From that number, across those 600 to 800 years, came 10 to 13 million living people. Waldman's ancient DNA study confirmed it independently. Two research teams, two approaches, the same number, 350. If you have Ashkenazi Jewish ancestry, you are the genomic consequence of those 350 people. If you have had your genome tested and seen elevated carrier flags for Tay-Sachs, for Gaucher, for BRCA, you are reading a document those 350 people wrote in their DNA during the worst decades of medieval European Jewish history. The science cannot tell you their names. It can tell you they were there. It can tell you the winter broke. It can tell you that, somehow, you are on the other side of it. If this changed something in how you understand where any of us actually come from, subscribe and follow The Inheritance Code, because the next episode goes somewhere older. And if you have someone in your life who carries this ancestry, or simply someone who thinks carefully about where human beings come from, send them this. The 4 women whose mitochondria became the inheritance of millions lived and died without knowing what they had done. They did not survive for history. They survived the way everyone survives, one decision at a time, in a city that did not particularly want them, in a body that had no idea how long it would have to last.